Hi, my name is Mehtab Singh. I'm doing PhD at McGill University in Montreal. We do gene editing in oats because Canadian winters are very hard on the oats, and we don't have good winter hardy oats that can be cultivated in the Canadian short agroclimatic conditions. So that's where my project comes in, and we use Plasmidsaurus at various levels of our CRISPR-Cas9 gene editing workflow. This is the workflow for gene editing, where it starts from plasmid construction. So once we have a guide RNA that is used for gene editing with the Cas9 protein, so we clone it into the gene editing construct that we have, but we really want to make sure if the guide is really inside the construct before we do all of this work that takes more than a year to achieve. So we use Plasmidsaurus here to confirm if the guide RNA is inside the construct and is ready to bombard and transform into the oat callus. And once we go through all the steps, again, we have our transgenic plants, and we want to sequence them. And oat is a hexaploid crop, so it has 3 different genomes, A, C, and D, and it makes it really hard to do it conventionally with Sanger sequencing. So we can design a primer that amplifies all the 3 copies of a gene. For example, here I have targeted VRN3 gene, which has 3 copies. So they're very similar to each other. We amplify the gene, all the 3 copies together, and we send it for deep sequencing, our premium PCR sequencing from Plasmidsaurus. And they give us all the 3 amplicons and the gene editing variants, providing us a really good coverage. And we are able to decode what happened with each copies of the gene with the gene editing construct. And it saves us a lot of time, and it's not really expensive. So once we have our gene edited lines, we have to phenotype them. So here you can see I have my wild type plant, which is not edited, is flowering beautifully, whereas my mutant are not flowering. So now I really want to know what happened at the genetic level and the gene expression level, why they are not flowering, and they're producing like different sorts of phenotypes. You can see here like broader leaves, longer leaves, thick stems, and whatnot. So now we are planning to use the new technology that is RNA-seq being offered by Plasmidsaurus, which is also at a really great price of just $50, and it gives you a great coverage, and you can send multiple biological replicates and different treatments, and hopefully we'll be publishing and will be good for adapting the oat to the Canadian short agroclimatic conditions, and so that you can continue to have your breakfast cereal irrespective of the cold weather in Canada. Thank you.